Good day, this is the Professopher, drwillphd, PhD. Today's Wednesday, January 18th, 2023. It is 5:25 and 21 seconds, a.m. Eastern Standard Time. And today's research, pancreatic cancer. So let's jump to it here. As stress-tolerant cells drive tumor initiation in pancreatic cancer. Date January 16, 2023. Source, University of California, San Diego. The findings may inspire new chemotherapeutic drugs targeting early stages of tumor formation and spread. As pancreatic cancer is one of the most lethal cancers, notoriously resistant to treatment, almost all patients experience cancer recurrence or metastasis. In the early stages of tumor formation, cancer cells, those with cancerous mutations, called oncogenes, experience a loss of adhesion to other cells and the extracellular matrix. The web of macromolecules that encase and supports all cells. This isolation leads to a local lack of oxygen and nutrients. Most cells do not survive such isolation stress, but a certain group of cells can. The tumor-initiating cells play a major role in the formation, recurrence, and metastatic spread of tumors. What sets them apart from other cancer cells is a resilience to these early, substandard conditions. The scientists say, like cacti in a desert, they can adapt to the harsh environment and set the scene for further tumor progression. The goal was to understand what special properties these tumor-initiating cells have and whether we can control the growth and spread of cancer by disrupting them. As subjected to pancreatic cell lines to various forms of stress, including low oxygen and sugar levels, they then identified which cells could adapt to the harsh conditions and observed which genes and molecules were modified in these cells. The researchers observed the stress-tolerant tumor-initiating cells showed reduced levels of tumor-suppressive microRNA MIR139-5P, and this in turn led to the upregulation of lysophosphatidic acid receptor 4, a G-protein-coupled receptor on the cell surface. The lysophosphatidic acid receptor 4 is not normally found on happy cells, but it gets turned on in stressful environments to help the cells survive, which is particularly advantageous for tumor-initiating cells. The researchers found that lysophosphatidic acid receptor 4 expression promoted the production of new extracellular matrix proteins, allowing solitary cancer cells to start building their own tumor-supporting microenvironment. The new extracellular matrix was particularly rich in fibronectin, a protein that binds to transmembrane receptors called integrins on surrounding cells. Once the integrins on these cells sense the fibronectin, they began signaling the cells to express their own tumor-initiating genes. And eventually, these other cells were recruited into the fibronectin matrix laid by the tumor-initiating cells, and the tumor started to form. The findings established a critical role for lysophosphatidic acid receptor 4 in pancreatic tumor initiation and a likely role in other epithelial cancers. As isolation stress is not the only way the signaling pathway can be triggered, the researchers say drugs are also designed to put cancer cells under stress. The research team found that treating cultured tumor cells and pancreatic tumors in mice with standard of care chemotherapeutics also led to the upregulation of lysophosphatidic acid receptor 4. The scientific researchers said this might explain how such tumor cells could develop a stress tolerance and resistance to the drugs. The further experiments also showed that using integrin antagonists to block cells' ability to utilize the fibronectin matrix reverts the stress tolerance benefit of lysophosphatidic acid receptor 4 expression. As thus, the researcher suggests targeting the lysophosphatic acid receptor 4 pathway or disrupting the fibronectin integrin interaction can be effective in preventing the growth, spread, and drug resistance of pancreatic tumors. We can think of tumor-initiating cells as being in a transient state that can be induced by different stressors, so the critical goal would be to prevent oncogenic cells from ever entering this state. And now that we've identified the pathway, we can assess all the different ways we can intervene. The researchers suggested a new drug targeting this pathway could be used as a prophylactic in patients at high risk of developing the disease, or to prevent new tumors from forming in cancer cases with a high likelihood of metastasis. And by pairing the new drug with existing chemotherapeutics that put stress on mature tumor cells can also mitigate the effects of drug resistance and make cancer treatments more effective. And by treating cancer can feel a little like whack-a-mole, but if we have 2 or 3 hammers and we know where the moles are going to pop up next, we can beat the game. And this is the Professopher, Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.